one of the two chemical senses. This is a sagittal slice of the nose. And what you'll see is that the nasal cavity here up, you have these olfactory receptors that is sitting here in the upper part of the nasal cavity. They project to these what's called glomeruli in the olfactory bulb. And you will see that different types of smells will activate different types of receptors and then be mapped onto these glomeruli. So there is, of course, a distribution in the nasal cavity of these receptors, but in the olfactory bulb you have these parts here where there is a collection of neurons that are responsible for specific smells. If you take a look on the human, you will see that you have the olfactory bulbs here. They are very small in humans compared to other animals. If we make a map, this is a map from salamander, you will actually see that if you map out the activity of the olfactory bulb, you will see how different types of smells will map onto these on the olfactory bulb and activate different parts of these glomeruli. And it's believed, although it's not proven directly, but at least that this pattern of activation in some way makes you able to distinguish between different types of smells. So because of these patterns, you can then combine these patterns in various different ways and then be able to smell differently. And the number of different receptor types is not like there is one receptor type for each smell, but you can combine maybe a set of receptor types and then combine them into different smells in combination. So you don't have to have one specific receptor for each specific smell type. You can combine, and then by combining these, you can code different smells. If we look at the processing, the central processing of smell, you have the olfactory bulb here, which projects to these olfactory nuclei and then up to the thalamus. And then signals are sent to the cortex, mainly the orbitofrontal cortex. So this is this part of cortex here, so the most inferior part of the medial part of the frontal cortex. It's called the orbitofrontal cortex. It's not only responsible for smell, it's also responsible for pleasure processing, etc. But to some extent also related to smell. And then also to the olfactory cortex and related regions in the temporal lobe. Taste. We have these taste buds on the tongue, which process different types of smells, so salt and bitter and sweet and acidic smells or tastes. And so even though we normally say that there is this different organization that parts of the tongue process either salt or sugar or bitter, you will see that large parts of the tongue can actually process different, or all of the different tastes. If we look at the central processing of taste, you will have pathways going from the tongue into the medulla, and then into the left gustatory nucleus, and then up to thalamus, and then into the primary gustatory cortex up here. So it's in the area where we also have the insula up here. Finally, I will just tell you a little bit about this man here, Ian Waterman, and I'm sure that you will later on hear a little about him, and some of you may already know him. But Ian Waterman is a Brit, and around his year when he was 20, he lost all or a lot of his sensation from the neck and below. And today I'll just briefly talk about something that's related to the next part of my talk today on EEG, and that's looking at brain activation related to stimulation of his peripheral nerve system. So this guy here, he has lost his large myelinated fibers, which means that these fibers in particular, they no longer function from the neck and below in him. So he cannot feel touch, he cannot feel movement of his body, but there is still some pain sensation and temperature sensation left in him. So actually, when the doctor spoke to him and tried to make tests of his ability to sense position of his limbs, he was actually able to sense some type of movement, for instance, of his shoulder, because he could feel that in his armpit the temperature got lower when it was lifted compared to when it was lowered. So the temperature sensations was to some extent still intact, but his ability to actually sense anything from the muscles and joints was lost. And if we then looked at measuring his brain activation, what they did was that they stimulated a nerve down in the hand, the median nerve. And normally you will see that that healthy subjects, they respond in the primary somatosensory cortex after approximately 20-25 milliseconds. But in the case with Ian, it took almost 80 milliseconds before there was a response in the brain. So suggesting that all the fast diameter axons, they couldn't process any stimulations, and it was only the small diameters and very slow fibers that could process any information to his primary somatosensory cortex. Furthermore, the stimulation intensities that were needed in order to activate or give a response in the brain was much higher than what you will see in normal subjects. So you had to increase the intensity of the stimulation up to 3 times. And again, suggesting that these large diameter fibers, which are easier to activate than the small diameter fibers, well, they were actually not working. So that's a way that he could actually be diagnosed with respect to which types of fibers that were not working in him. So I think that was what I wanted to tell you today. And yeah, I think we should have a break now. Thank you.